Requirement of hippocampal neurogenesis for the behavioral effects of antidepressants. Modern treatment of depression is based on the monoaminergic hypothesis of depression, which states that depression is caused by decreased levels of noradrenaline and serotonin. Pharmaceutical treatment involves drugs that increase the amount of these neurotransmitters, but not all drugs that do this also relieve the behavioral symptoms of depression. The physiological effects of depression and depressive symptoms include atrophy, dendritic shrinkage, and decreased hippocampal granule cell neurogenesis. The decreased neurogenesis has been observed to increase when a depressed patient is put on antidepressant medication. The treatments given to the mice include 3 common antidepressants and an antipsychotic agent. The antidepressants come from 2 families, with 1 SSRI and 2 tricyclics. Because only increasing neurotransmitter levels does not relieve symptoms, and because adult hippocampal neurogenesis decreases with stress and increases with antidepressant treatment, we hypothesize that adult hippocampal neurogenesis is necessary for effective treatment of depression.
We used an adaptive novelty-suppressed feeding test in which we treated mice for either 5 or 28 days before assessing latency to feed. The mice were immediately returned to their home cages after testing, and their food consumption over a 5-minute period was measured. The effects of fluoxetine were determined by treating adult mice with either vehicle or fluoxetine. They were then injected with BRDU on the final day of treatment and killed 24 hours later. To compare the effects of serotonin and norepinephrine enhancing antidepressants in wild-type mice and in mice lacking the serotonin 1A receptor knockout, adult mice were treated with fluoxetine, imipramine, decipromine, and vehicle for 28 days before being given the NSF test. A serotonin 1A agonist was administered for 28 days prior to the test in order to determine if the activation of serotonin 1A receptors is sufficient to alter NSF behavior. Wild-type and knockout mice were injected with BRDU after a 28-day treatment with fluoxetine, imipramine, or vehicle. One group was killed after 24 hours and the second group was killed 28 days after BRDU injection. Low doses of X-rays were delivered to the hippocampus as well as a region just rostral and one just caudal to the hippocampus to determine if the focal irradiation produces similar effects as antidepressants and to determine if those effects are specific to the hippocampus. Time results in deterioration of the state of the mouse's coat. Sham and irradiated mice were subjected to CUS. Fluoxetine was administered to half of the mice in the group. At the end of the period, each of the mouse's fur was assessed and assigned a score.
The results of this study show that hippocampal neurogenesis is related to the reversal of depression-related symptoms from antidepressants. When given a variety of antidepressants, fluoxetine, imipramine, and desipramine, the mice reacted to all 3 within 5 days, showing increased food consumption correlating to less depressed behavior. After 27 days, all antidepressants showed increased eating habits, while haloperidol did not. The delayed effect is similar to the delayed onset of antidepressant reaction in the human brain. The study also showed that fluoxetine caused a 60% increase in the number of BRDU positive cells in the dentate gyrus of mice, which were treated for 11 or 28 days. However, fluoxetine had no effect after only 5 days. It was also determined that the 5-HT1A receptors are required for fluoxetine-induced hippocampal neurogenesis.
Mice are less likely to feed when there's chronic treatment with antidepressants that have serotonergic or noradrenergic mechanisms, and also when there's chronic activation of 5-HT1A receptors. Mice that lacked the 5-HT1A receptors responded to tricyclic antidepressants, but not to serotonin selective reuptake inhibitors. This indicates that SSRI pathways rely heavily on the 5-HT1A receptors. When 5-HT1A receptors are eliminated, there is a blockade of behavior and neurogenic effects of fluoxetine. This indicates that the processes may be related, however, they are dissociated in 2 instances. One is in the NSF test. The knockout mice show high levels of anxiety but normal levels of basal neurogenesis, and the other occurs when there is 28-day ablation of neurogenesis, but there is no behavioral effects. The hippocampus has been known to function in memory and learning, but newer evidence is showing that it can also function in the modulation of emotional responses. Some of this evidence is that the lesions on the ventral hippocampus or administering a drug targeting the ventral hippocampus results in altered behavior of anxiety-prone mice. There's also been a double dissociation and the role of ventral and dorsal hippocampus in spatial learning versus hyponeophasia. When there is a dorsal lesion, spatial learning is affected but not hyponeophasia. But when there is a ventral lesion, there's decreased hyponeophasia but no effect on the learning.
Future research could consist of using adult female mice rather than male between the same age range of 12 to 20 weeks to compare and see the differences. Other improvements in the experiment could be to have less number of mice per cage in order to reduce the amount of stress that the mice put on each other. Stressors were controlled within the experiment with things such as cage tilting and predator sounds, but the stress that the mice put on each other was not accounted for. Another possible improvement would be to find a way to remove or account for an independent source in the knockout mice that gave them higher levels of anxiety-related behaviors. And lastly, rather than using a selective serotonin reuptake inhibitor in the two tricyclic antidepressants, we could use a monoamine oxidase inhibitor or atypical antidepressants.